Hi guys. So we're going to start today by reviewing what we talked about with proteins and DNA. So last week we talked about how DNA has genes that actually code for proteins. Okay, and you don't have this page in your packet, you're just listening. And so we have a bunch of different types of proteins in the cell, and here are just some examples that are important to remember. Remember that enzymes are proteins, right? Cell membrane receptors are proteins, insulin, other hormones, right? And antibodies, hemoglobin, which transports oxygen in your blood, and then your traits themselves, when you look in the mirror, are actually formed by proteins. Okay. And so let's talk about how is DNA actually used to make proteins? How does that happen? Right? When the cell makes proteins, this is called protein synthesis. So remember, synthesis means to make, right? And so there's this process to make proteins called protein synthesis, and that's what we're exploring today. Protein synthesis occurs in multiple places in the cell. It's going to start in the nucleus, okay, where the DNA sequence for a specific protein is copied and then taken to the ribosome by an enzyme. The ribosome will read the sequence and find the amino acids that correlate to the DNA sequence to build a protein. We're going to highlight nucleus here and ribosome. Okay, that sentence, you may not know exactly what's going on, but I want you to know where in the cell protein synthesis takes place. It's going to take place in two steps in two different places. Okay, so let's go ahead and start really breaking down how does this happen. Here's some quick facts before we get started, like an overview. Okay, protein synthesis is a two-part process. Okay, the first process is transcription. The second process is translation, and it occurs in the nucleus and ribosome. Okay, so the transcription occurs in the nucleus, and the translation will occur in the ribosome. So let's talk about this word transcription. Okay, and what happens. Okay, so this is when we're going to take the DNA and turn it into mRNA. Okay, so let's talk about this. I'm going to give an analogy. Okay, remember how I said that DNA is a recipe book, right? And I want you to picture that your grandparents have this family recipe book and your grandma has it locked up so that nobody takes it out. And let's say you don't have a cell phone, right? But you want to take a recipe from the recipe book to take it home and do it. So what you might do is you might take a piece of paper and you might copy the recipe down, right? So then when you leave the house, you have a copy of that recipe. It's the original recipe, you just made a copy of it, right? So that you could take it home and then make that recipe on your own without taking the recipe book out of that house, right? This is the exact same thing that happens in the cell, right? The DNA contains the recipe for building specific proteins. But here's the issue. DNA cannot leave the nucleus ever. Okay, it can't leave the nucleus ever, 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 ever. DNA has to stay in there. So the question is, how do we get that code for the protein to the ribosome where the protein is made, right? Well, we have this beautiful thing called RNA polymerase, and it's an enzyme. Okay, and so what RNA polymerase does is really, really cool. It's going to come into the nucleus, okay, and it's going to find the gene that it's looking for, and it's going to make a copy of it. Okay, when it makes a copy of it, it's going to make a copy of it to it's going to make an RNA copy of it. So RNA is a little bit different. So when it copies it, right, it's going to copy it not into DNA, but into something called RNA. Okay, so I want you to think about it like maybe in the recipe book is written in pen, right? But then when you copy it down, maybe you copy it down with marker or with pencil. So it's the same thing, but you're kind of using a different utensil. So when RNA polymerase comes in, it's going to find the gene that it's looking for. It's going to make a copy of the DNA, very similar to you writing down, the same exact recipe. And that is going to be in the form of RNA. Okay, that RNA will actually head out of the nucleus into the ribosome for the next step. But let's take a moment and actually see what this looks like and how you might be asked about this. Okay, let's take a moment first and explore RNA itself, okay, as a molecule. Because once again, transcription is taking DNA and making a copy of it in the form of RNA. So let's label these two pictures. Let's call this DNA and this is RNA. Some differences you may notice already is that DNA has this kind of a double strand here, like two strands, right? And two bases right here. Whereas RNA is just one strand. Okay, so it's a lot smaller. Okay, so let's talk about some differences. Like we just said, a lot smaller. That is really important. RNA is a lot smaller. It's single stranded. Okay. RNA is similar because it's still made of nucleotides. Okay, so it's still made of nucleotides. And it's still a nucleic acid. So those are the similarities. There's really just two differences, honestly, or three kind of big differences. One is the sugar that RNA has. Okay, it's going to have ribose sugar, and DNA has deoxyribose. It's not that big of a deal, and you really won't be asked about that. But that's why it's called RNA instead of DNA. Okay, so it has a different sugar. It's smaller. It's single stranded. Okay. And here's the major difference that matters to you guys. It's that DNA has four bases, right? It has adenine, guanine, cytosine, and thymine. A's and T's, C's and G's. RNA is just a little confusing, okay? Because it has adenine and guanine and cytosine. However, it does not have thymine. It has this nucleotide base called uracil or U. Thymine cannot occur in RNA. It just has to do with the chemistry of it. RNA just can't have thymine. Okay. And so instead of A and T, okay, it's actually A and U. So uracil is complementary to adenine in, I meant to say RNA. Okay. So it just takes the place of T. So let's look at our pairing rules for when you're going from DNA to RNA, right? So when RNA is making a copy of the DNA sequence, this is how it does it, right? So let's go ahead and take a look. So we have in RNA, right? C pairs with G, same as DNA. G pairs with C, same as in DNA. You can still say cars and garage. Here's the difference. If DNA has an A, then it's going to pair with a U. Don't pay attention to that box, okay? This is our base pairing rules. So I like to say apples and umbrellas. I really don't know why. I just kind of feel like it's weird, and I kind of feel like I remember it that way. So C pairs with G, G pairs with C, and A pairs with U. This actually meant to say T pairs with A. So if this is DNA up here, please write this. This is the RNA that pairs with it. Okay. So here's the confusing part. I just want you to think T still pairs with A. Okay. Trees on apples. Okay. So if you see a T in DNA, RNA will have an A. The issue is if you see an A in DNA, you get a U. So what I typically do is when I do the pairing, if I see a T, I just do the normal pairing. And then every time I see a T or I'm about to write a T, I just change it to a U. So let's go, we'll get a little bit of practice in a moment. All right, so I just want you to show you an example first. So once again, this is transcription, okay? Because we're going from DNA to RNA, that is transcription. So let's take a look at this example here. So I'm going to go ahead and use my laser and just show you guys. So in DNA, it goes A's and T's, C's and G's. In RNA, you will see a pairs with U and T pairs with A. So let's take a look. Okay, we see a G, we get a C. We see a G, we get a C. We see a C, we get a G, because G's and C's still pair together. Now watch. If DNA has a T, we put an A, because A exists in RNA. We see a T, we put an A, because adenine exists in RNA. Here's the only difference right here. And let's go ahead and highlight that. When DNA has an A, RNA is going to put a U, because it doesn't have T, but it has every other letter. Okay? So I want you guys to go ahead and answer these questions. You can pause now or answer them later, but I want to walk you through, actually, I'm not going to walk you through the practice. You guys can go ahead and do that on your own and ask any questions. Remember to look at the base pairing rules. And then this is a really good example up here to help you out because it has every letter. So you can kind of use that to help you as you do some practice, okay? Okay, let's talk about the second process called translation. Okay, so that's the second process. Okay. And let's just review transcription really quickly because it just is a little confusing. So I like to repeat myself so that it makes sense, right? So let's give a scenario this time. Let's say that your cells, you are eating, like let's say you just were eating breakfast, right? And so your cells need to make some enzymes because you're eating. So what your cell is going to do, right? Your stomach cells need to start making enzymes. So your brain will tell your cells, hey, we're eating, make lactase enzyme. So your cells, okay, will send RNA polymerase to go find the gene for lactase enzyme and to start making copies of it. Okay, that happens in the nucleus. That's actually transcription, right? When RNA polymerase is going to go find the gene for lactase enzyme, it's going to start making copies of it. Once it has the copy, once it's copied it from DNA to RNA, that RNA will leave the nucleus, okay, and it will leave the nucleus and it will go to the ribosome so that the ribosome can start building lactase and send it out of the cell to the stomach. All right, cool. So just to review transcription. Transcription is DNA to RNA. It happens in the nucleus. It's when we take DNA and copy it to RNA. So let's look at this picture. This is showing the RNA strand leaving the nucleus, right? Leaving the nucleus, and it's going to head to the ribosome next. Okay. Once mRNA is in the ribosome, what the ribosome is really cool at doing is it will read the code and grab the amino acids that the mRNA codes for. Okay, so let's just take a look at this picture really quick. Okay, what we see here is we see this ribosome. Okay, here's the mRNA. Okay, that's the mRNA. It's getting fed through the ribosome like this. And every 3 bases, okay, see this right here? Every 3 bases codes for 1 amino acid. So if you look, these guys right here, what they're doing is they're grabbing amino acids and they're stringing them. They're kind of reading this code right here and seeing which one they need to grab, right? Is it red, green, orange, blue? Which one is it? And then these little guys kind of string on and make this amino acid chain. Those amino acids are the building blocks of proteins. So that will build the protein that you're looking for. Okay. So every 3 letters of RNA is a codon. Okay, so the sequence above has 3 codons. So what we're going to do to figure out how many codons we have is just put a line. So it's just every 3 letters, right? So it's like this, look, UGC, right? AAU, CGG. So there are 3 codons there. Usually there's way more codons in a gene, but we're just simplifying it, right? So once again, each codon, every 3 letters codes for 1 amino acid, and there's 20 different amino acids. Okay, And depending on the combination of amino acids, when they're sequenced differently, so for example, right here, okay, we have an amino acid sequence that has that is different, right, than this one, and it's also different than this one. It has different amino acids in it. So each protein will be different depending on the sequence of amino acids. So the sequence of amino acids will make different shapes in thousands and thousands of different proteins, even though we only have 20 amino acids, because they're kind of put together in a different sequence, right? So if you have different types of Legos in different colors, you had 20 of them. You would take those 20 and you could put them in different orders, right? And build kind of different things and different looking structures with that. It's the same thing. Okay, so let's actually practice translation. So what I want you guys to do is I want you to label this as translation. I'm sorry, my bad. So this is transcription right here. How do we know? We're going from DNA to RNA. Okay, that's transcription. And this is translation. Translation is when we go from RNA to amino acids. Okay, so let's go ahead and get our first practice. Okay, look, we're going to read the bases. We have 3 codons here, okay? So remember how I said we have 3 codons. So in RNA, we kind of just separate every 3, right? So here's this codon right here, UGC, that's the first one. It's down here. AAU, we put it down here, and then CGG, okay? So we have our 3 amino acids. We just need to figure out what amino acid, or I'm sorry, 3 codons. We need to figure out what amino acids they code for. So here's how you're going to do it. All right, UGC is the first one. Okay, UGC is the first one. So if the first letter is U, we're going to start right here. And usually I take my finger and I kind of do this. So it's UGC. So we're going to go U, and I go to G, right? And then I go to C right here. So this amino acid is cytosine. Cysteine. So we're going to put CYS. And we're going to put in parentheses the full name. Because usually we abbreviate it. So how did I know that was the abbreviation? I just looked right here. Once again, all I did was it's UGC, right? U, so I started in U, G, C, and it falls in this in the cis category, right? So I'm going to do the next one, AAU. So AAU, that is ASN, or asparagine. And these are all the 20 amino acids right here. These are names of amino acids, okay? So CGG, we start in C, CG, that is proline or pro. So you're doing translation right now. This is what the ribosome does. It reads that RNA and it spits out the amino acids that it codes for to build a full protein. At this point, you guys are just going to finish on and get some practice in. Reach out with any questions.